Okay, this tutorial is about fish gas exchange. Fish have gills as their gas exchange surface, and the gills have certain adaptations that allow the gases to be exchanged as quickly as possible, and that's what this tutorial is about. The oxygen has to diffuse from the water into the blood where it's picked up by a red blood cell, and then carbon dioxide, which has been given off through respiration by the fish's cells, enters the blood and has to diffuse from the blood to the water. So the fish has 8 gill arches, and each gill arch, 4 on each side of the mouth, looks a little bit like one of these. There's the arch sort of shape, and we've got this kind of bony structure here, and then layers of gill filaments. So these are very, very tightly packed, a little bit like the teeth on a comb, really, really tightly packed and layered up. And there's another section over here. So instead of having one big lump of tissue, if you like, to have one gas exchange surface here, they've been subdivided. So it has been increased the surface area. Okay, so we can say that each gill has many filaments, and that's one of the things that increases the surface area. But there's something else that increases the surface area, because each of the filaments has many lamellae, and I've got another little picture to show what they look like. So if I just reveal one filament enlarged, there we go, and you can see that if you were to look at the filament in really, really close up detail, you'd be able to see that it's got these kind of little sideways shelf-like structures at right angles to each filament, and these are called lamellae. So let's label that. So you can see that what's happening is you've got all of these different layers, these layers of filaments increasing the surface area, and then each filament has many lamellae. So these would be these extra levels of surface area on each filament. Okay. So let's write something about the lamellae. Inside each lamella are many blood capillaries. So the oxygen, let's imagine that this is the water around the gill filament, around the fish's gills. The oxygen has to diffuse from the water across the wall of the lamella and then into, I'm just going to have to imagine that they're there, into the capillaries. Okay. And likewise, the carbon dioxide has to diffuse from the blood capillaries out into the water. So we can make another observation here. The capillary and the lamellae walls are very, very close to each other, which obviously limits and minimises the distance that the gases have to travel from the blood to the water and from the water to the blood. But also, the walls of these structures are very thin. They're made up of a single layer of cells. Okay, so if we go back to our list up here, we can see that we can assign a number 1 for large surface area to this detail here. So if you ever had to answer a question about how the gills are adapted to maximise surface area, you would have to mention the filaments, but also the lamellae. And that's pretty much what you'd have to write if you were answering a question about that. And then if you were trying to answer a question about how the gills minimize the diffusion pathway, you could say something along these lines. Yeah, the lamellae walls are right next to the capillary walls, and also they're both made up of a single layer of cells, so the gases don't have to diffuse very far. So we're left with the concentration gradient. So how do we always ensure that there's always more oxygen in the water than there is in the blood, more oxygen in the water than there is in the blood, so that there's always oxygen diffusing in the correct direction from the water to the blood? So there's this system. I've got another little picture here. There's a system called the countercurrent system, which I'll try to describe to you now. So what we've got here, if we kind of think about what's inside the lamellae. Now this is very diagrammatic, but it kind of gives you an idea. So we've got the lamellae walls here and here, and then inside the lamellae we've got the blood capillary, the walls of the blood capillaries. You can see they're really close to one another, and they're both just made up of a single layer of cells. And we can see that the blood will be flowing in a particular direction. So the flow of blood, let's say it's going from right to left in this case. But the important thing is that the flow of blood is in the opposite direction to the flow of water outside the lamellae. So the water is flowing in this direction, the opposite direction. And as it does that, the oxygen is able to diffuse into the bloodstream and be picked up by the red blood cells. Okay, so I'm going to write down a couple of things about why this is so important. Why, let's just highlight this arrow a little bit more, why this flow, this countercurrent flow is so important. And then I've got another little diagram to show you to finish the tutorial. So let's say if you ever had to talk about this system, you would say something along the lines of the water outside the lamellae flows in the opposite direction to the blood inside the capillary. And this is called the counter current flow. Okay, current referring to the flow of blood and the flow of water, and counter meaning they're going against each other, they're occurring in the opposite directions. Okay, and then let's also write down what this means. What does this mean in terms of the concentration of oxygen in the blood, in the water, and the concentration of oxygen in the blood? So this countercurrent flow, ensures. Let's make sure that you can see that that's the word this. This ensures that blood is always next to water with a higher concentration of oxygen. Okay, now there's actually one more bullet point that I want to add to this, but let me just show you a little diagram to try and illustrate this idea here. Okay, so here's something that I put together earlier. Let's have a look at this one first of all. This is the scenario as if the water was flowing in one direction and the blood was flowing in the same direction. So this is the same current. This is not what happens, but let's just think about what it would be if it did happen. So here we have the percentages of oxygen in the water and the percentages of oxygen in the blood. So at the moment we've got 100% oxygen in the water and nothing in the blood. And of course, that represents a really steep concentration gradient. And so oxygen will be able to diffuse from the water into the blood. Okay. And then if we go a little bit further along, because some of the oxygen has now diffused into the blood, there's a little bit less oxygen in the water, but there's a little bit more oxygen in the blood, but there's still quite a big difference between the two, and so oxygen can still diffuse from the water into the blood. But now when we move a little bit further along, let's imagine that this is the whole length of the gill filament, if you like. Now we move a little bit further along, and now because a little bit more oxygen is diffused into the blood from the previous point of the lamellae, then there isn't a concentration gradient. So I shouldn't actually be drawing an arrow there, because we have now reached an equilibrium. So actually diffusion stops at that point because an equilibrium has been achieved. So let's write that down. Equilibrium has been reached. So diffusion stops. Remember, you can only have diffusion if there is a difference in concentration, like here, but not here. Okay. So this is what happens if you have the blood and the water flowing in the same direction. We would reach an equilibrium quite quickly. Let's now have a look and see what would happen if the blood and the water were flowing in the opposite direction. So this is counter current. This is what happens. Counter current. Okay, so the water is flowing in that direction. So it's starting here and ending at that end of the gill. And the blood is starting here and ending at that end of the gill. Okay, so if we start at this end in the water, we have 100% oxygen. Okay, and if we look at the percentage of oxygen that's in the blood, we can see that there is actually quite a lot of oxygen in the blood already, because it's already received some as it was flowing from that end to this end. But there is still a difference. There is still more oxygen in the water than there is in the blood. And so we have a concentration difference. The oxygen can flow in the correct direction from the water into the blood. And the same thing here. Okay, so you can see that there is always a very small difference. Might be small, but it's big enough. And there's always more oxygen in the water than there is in the blood until we get to this bit here. Okay, so we have not achieved an equilibrium until the very end. Not reached. And so diffusion can occur. Now, this is the important bit. This is the bit that you've got to include if you answer a question about fish gas exchange. We can say that diffusion occurs along the whole length of the gill. Okay, whereas here, when they were flowing in the same direction, it stopped, diffusion stopped halfway across. That's really inefficient. This is making the most of the gas exchange system and extracting as much oxygen from the water to the blood. Okay, so let's add this point here to our bullet points and then we're finished. Okay, so if we just go back to this, and we're going to finish off our bullet points here by adding, so diffusion of oxygen from water to blood occurs over the whole length of the gill. Really important to remember to add that last point. Okay, so if we just finish off by looking back at our little list of aspects of Fick's law that the exchange system has to meet, and we've just talked about the countercurrent flow here, which is to do with maximising the concentration gradient, so we can add a number 2 there to refer back to our list.